Good afternoon to everyone. My name is Umberto Maccio. I am a pathologist. Before starting, I would like to thank the organizing committee for inviting me. As pathologist, I am a sort of white fly at this congress, but I think it's important that specialists dealing with infectious disease know the limits and the possibility of diagnosis of infectious disorder in a pathology lab. So we will discuss in this short talk about routine practice and emergency aspects with some practical examples. Before starting, I have no disclosure statement, and let's go. I will show you here this beautiful picture of a bucolic Swiss landscape, and we will start with the first interactive questions. If you recognize that, I give you a few seconds to answer. And of course, the answer is full capacity. If you have seen the beautiful movie of James Bond, you will recognize this. So we will follow the same path, and we will start with a discussion of morphology-based diagnosis. Then we will move to the diagnosis based on histochemical and spatial stain. Then we'll discuss the possibility of PCR-based diagnosis on formalin-fixed paraffin-embedded material through molecular pathology, molecular techniques. And then we will shortly discuss an upcoming topic of metagenomics. So let's go with the first example. We have here a biopsy of the duodenum. And as you can see here, we have no atrophy of the villi, we have no intraepithelial lymphocytosis, you have no inflammation, so you may think it is a normal biopsy, but you have always to look carefully in anatomical pathology at details. And as you can see here, between the villi, you can see some structures here, which may be unicellular organism, and at greater magnification, you can see this pear-shaped morphology, and this is a diagnosis that we can do just relying off H&E section without performing other examination. My question is, what is your diagnosis? I'll give you some second to think about that. And the answer is, of course, Giardia lamblia. So it's something that we can diagnose just on duodenal biopsy. Another example, we have here a specimen of an appendectomy with purulent exudation here, suspicious for acute appendicitis, and we can see here in histopathological exam a transmural infection. And also going to details, we can see here some structure with a membrane, with some hooks, with eggs inside, and with part of intestine. So what is here your diagnosis? I give you here also some second to think about that. And the answer is B, acute appendicitis with enterobius vermicularis. So these are just two cases to show that we are sometimes able to recognize immediately these bugs and to diagnose an infectious disorder. Sometimes this is not enough. Morphology is not sufficient to do such a diagnosis. And sometimes we have nuclear changes, inflammatory pattern, or presence of microorganisms which may suggest a particular diagnosis, but are not specific. For example, in nuclear changes, you may find cautery bodies, for example here, in which you have the 3Ms multinucleation marginations of chromatin and molding, which is suspicious of herpes simplex infection. You have cautery B, which may be specific, suggestive of cytomegaloinfectious. You have molluscum body. You may have coilocytes, which may suggest an HPV infection. You may have negri body, found especially at autopsy in a Purkinje, which may be suspicious for rabies infection. And then you may see sometimes in hepatocytes councilonal body, which may be seen by yellow fever or by hemorrhagic fever like Ebola or virus Marburg. So in these cases, morphology is not enough. We need something more to diagnose these disorders. Another example is when you deal with necrotizing granulomatous inflammation, which is suggestive of mycobacterial infections. You may not distinguish between mycobacterial type just relying on inflammation, or it is consistent with another kind of inflammation. We don't know. We have to perform other techniques. Or lymph nodes, when you have these beautiful trials of follicular hyperplasia, B-cell hyperplasia, and epithelial histiocytes in paracortex, like here, which is suspicious of toxoplasmosis, but of course also here is not diagnosed, it is suggestive. Or pseudomembranous colitis, again with the other example, you may also have presence of microorganisms which are not specific. For example, you may find some bugs here in these histiocytes. You don't know if this is leishmaniasis or histoplasmosis. Or also if you find some kind of acid-fast bacilli in special stains, like for example Zillnissen here, you don't know if it is a Mycobacterium tuberculosis or other kind of mycobacteria. So in this case, you may need other examination. I will show here an example of it works, for example, with immunohistochemistry. We have here a case of a patient with dysphagia and weight loss. In endoscopy, they found a lesion in distal esophagus, which was suspicious for esophageal carcinoma. So those are the two biopsies that we got, and we can see here in this part a desmoplastic stroma with typical cells with squamous differentiation consistent with squamous cell carcinoma. So we have the diagnosis, you may think, but you have also to look carefully to other part of the biopsy. And here, for example, you can see these cells with these inclusions here with big nuclei, which are suggestive of Cowdry B bodies. So we may also have parallel to the carcinoma CMV infection. And indeed, we confirmed that with a stain for cytomegalovirus, the presence of cytomegalovirus virus. So we have combined esophageal carcinoma plus cytomegalovirus esophagitis. So it is important because, of course, esophageal carcinoma is most important for the treatment, for the prognosis, but you have also to treat the esophagitis. Another example, you have here a patient with cough and weight loss, and in CT scan you see a suspicious nodule of 8 millimeters. So the surgeon sent us for a frozen section to rule out a tumor, and that was what we have seen. So we can see here a lesion with central necrosis, and in the peripheral part with some kind of inflammation. If we go to a greater magnification, we can see epithelioid histiocytes with a giant cell of Langerhans type, and here we can see the necrosis inside. So it is not a tumor. This was my response to the surgeon. This was a necrotizing granulomatous inflammation, and this pattern is suggestive of infectious etiology, especially Mycobacteria. In these cases, we should always send to microbiology lab some fresh tissue in order to perform some cultures of other PCR analysis. We performed a Zillnissen stain, which was negative for acid-fast microbacteria, and we performed also some fungal stain. For example, here is crocod stain, and we can see ears and bugs of about 2 to 3 micrometers. And what is here your diagnosis? What do you think? Well, we have here, for example, a morphology which is consistent with Histoplasma capsulatum, and this hypothesis was confirmed by the microbiology lab with panfungal PCR. So fungal diagnosis is not so easy in anatomical pathology. Basically, as you can see in this nice paper published some years ago, but still important, we control the dimension of the fungi and also the morphology. Combining these two aspects, we are almost always able to suggest a diagnosis, but you may need to confirm this by a microbiology lab through panfungal pan-PCR. So I will move then to other techniques, especially to immunohistochemistry again, and then to molecular pathology. Also with an example. Here we have a cervical biopsy. We can see here in the upper part of the lesion coilocytes with this halo and with this racinoid nuclei, which is suspicious of HIV infection. In this case, we will diagnose a low-grade dysplasia based on morphology. So we will call this LCL CN1, so cervical intraepithelial neoplasia type 1. So it is sometimes difficult to distinguish between low grade and high grade. In these cases, you may rely on P16 immunohistochemistry, which is overexpressed in case of high grade dysplasia. In the cervix, it's not a surrogate marker of HPV, like in other locations, for example, a tumor of head and neck part, in this case you may have an upregulation of P16 in case of high-grade dysplasia. Sometimes you may need to know if you are dealing with an infection of HPV low risk or high risk, which is important for the management of the patient, and you may have in this case an in situ hybridization. In situ hybridization is an RNA-based technique in which you have a cocktail of many sons which binds with low risk or high risk HPV, depending on which kind of HPV you are searching for. In this case was a low risk HPV in situ hybridization. Sometimes, and which is positive of course, so we are dealing with an HPV low risk infection. Sometimes you may need to know what kind of HPV type is that. So in this case you are going through PCR based analysis. And here you can see how it works. You extract DNA, and then you can see here several samples of different patients, and you have always to control that you have a positive control here. Positive control, you have an housekeeping gene, versica. As you can see here, is correctly amplified, showing that DNA quality is good, so you have always to control that. And then you have the amplification of these genes here of HPV, which are obtained through specific primer for these genes. So when you analyze the other samples of other patients, you can see also that they have a good amplification of versican in every case, positive and negative. And in positive cases, you can see here they do have some DNA which is present in these genes. So you have for sure an HPV infection or HPV DNA in this sample, but you may want to know what kind of HPV type is this. So you can sequence this DNA here, and you put your sequence that you obtain through Sanger sequencing in this tool BLAST, basic local alignment searching tool. You just give your nucleotide sequence, and then BLAST contains genomes of many microorganisms, including viruses, bacteria, etc., and perform a matching of this sequence with your sample. So you may give here the response, and here, for example, we have in this specific case, this example, human papillomavirus type 16. So this is quite reliable, quite sensitive. It's enough to have one positive cell per 7,500 cells, so quite a sensitive technique. Sometimes you do not know if you are dealing with a specific virus, so you may want to ask for a panel, and this is not done in every pathology lab. For example, this is in a reference laboratory of infectious pathology in Switzerland, in Listal, and as you can see here, you can choose from different PCR-specific bacterial analysis, or you can also order a panel. For example, if you have a respiratory infection in a lung specimen, formalin fixed, paraffin embedded, it works as well, and you have an amplification of DNA through this primer for all these genes. So you may have your answer if you don't know with which microorganism you are dealing with. The problem is, of course, if you allow me a metaphor from the astronomy world. Currently, we are working with molecular pathology, and this is what we know. But this is what we have still to explore, so there's a lot more. In our praxis, we are able to recognize microorganisms, we are able to match them with a specific clinical picture, so we are doing good diagnostic, but there is something more that we will need to approach in the future. We will need to study microbiomes, microbial abundance, functional characterization, screening for reservoirs, antibiotic resistance. They are all topics which are emerging and that we are not able to address based on our current technique. So an answer to this problem may be with metagenomics. Metagenomics is a study of genetic material coming from clinical or environmental samples, which enable the analysis of collective genomes of microorganisms. You need no culture, And I should remember you that we are able to culture just 1% of all bacteria existing. So this is very important to keep in mind. And we have basically two approaches, targeted or untargeted. What is the difference? In targeted metagenomics, we will amplify just specific gene of a class of microorganism. For example, we will amplify a specific gene which is present in all bacteria, but has some difference among bacteria. So we may distinguish the bacteria which are present in our sample, and we may be able to quantify them. The same may happen with fungi, which have some similar genes to bacteria and that can undergo the same process described. The problem is when you are not knowing if you are dealing just with bacteria, with fungi, or with viruses which have no specific gene in this context of target metagenomics. And so you may want to amplify your whole sample to control which kind of bacteria, fungi, virus are present. And this is the approach of untargeted or shotgun metagenomics. Let's show in detail what is going on with target metagenomics. So as I have said, you amplify a specific gene. An ideal marker gene is a universal and highly conserved, but have also variable regions. And you have primers that are able to amplify equally and exhaustively different targeted groups. As you can see here with the example of the gene for rRNA16S, you have some conserved regions which you can use as a primer, target for your primer, and you have variable regions. So the analysis of these variable regions here allows you to distinguish between the different bacterial types present in your sample. And the same may happen with eukaryotes such as fungi, but in this case you are using other genes. So this is the pipeline. You extract DNA, you do fragmentation, and you amplify these genes. Then you get this FASTA QC file in which you can also perform a quality control, and then you can use some tools online freely available like BLAST, for example, and you get a match between your sequence and your match between the genome present in the database and your sample. So the pros, of course, is cheap and is not so difficult in bioinformatical tools, but of course you have some problems. The main problem is that you have no consensus of primers, so you do not know if you are amplifying correctly these genes, and you have no information of virus because you have no specific gene for viruses, and you just get information of which species are present in abundance. This problem may be solved with shotgun metagenomics, in which you amplify all the DNA. And of course, it's much more complicated bioinformatically. You can use several tools, but you get much more answer. For example, you may also have some functional profiling, quantification, and whatever. But of course, it's much more expensive and much more complicated in the bioinformatic pipelines. So potential applications of course, not only the diagnosis and the detection of virulence determinants, but also microbiome analysis and oncology application. This is medicine, but of course you may have also application of metagenomics in other fields, like for example food nutrition, agriculture, etc. So it's really an upcoming tool. So I come to my conclusion. So from the point of view of a pathology, of course, we can recognize in histopathology some microorganism. We are able to diagnose them. If it's not sufficient in morphology, we can rely on immunohistochemistry or molecular analysis. But of course, we have to explore this upcoming tool of metagenomics, which have many possibilities, still many limitations, but I have to remember you, metagenomics is not yet validated for routine clinical practice. So we will need several studies to test the performance and the possibility of this tool. I thank you very much for your attention. This is my email address if you want to share cases or to ask for cooperation. I'm always happy to do that. Thank you very much again.